We're looking at parasitic protozoa. So on last week, we highlighted some very basic characteristics of protozoan organisms that you should remember. We talked about them being eukaryotic organisms, unicellular. They typically lack a cell wall. They have various modes of reproduction, but we're looking at asexual reproduction is what we see most in them. We're interested in those that are parasitic in nature, and we're going to take a look today at some other categorizations of parasitic protozoa. Last week, we looked at those that we can identify based on their mode of locomotion, right? So we talked about the amoebas, we talked about the ciliates, and then we talked about the flatulates. We're going to expand our conversation a little bit today and talk about another group. Oh, we talked about identifying parasitic protozoa based on those that are ingested, all right, and the two morphological forms that we can find those in. So today, you did have a homework assignment. And today we are going to look briefly. So our recapitulation was that we know that protozoans can be broadly classified based on their mode of locomotion, right? So the amoebas are using those pseudopodia, and then we have cilia and flatulates, where the flatulates are characterized by the presence of one or more flagella. And then the ciliates contain those small, thin, hair-like projections protruding from their body, all right? Today, we're going to look at some special types of flagellated protozoans, all right? And these groups are called Kinetoplastids, all right? Kinetoplastids are a type of parasitic protozoans that we look at. These are flagellated protozoans that are basically characterized by the presence of a large mass of DNA, all right? And we call that region a kinetoplast, okay? Jot that term kinetoplast down in your notes, kinetoplast, all right? Kinetoplastids are flagellated protozoans that are distinguished by the presence of a DNA-containing region known as a kinetoplast, right? So it's just a big, large mass of DNA, all right? And so we've been able to, you know, advances in technology, particularly DNA sequencing, has allowed us to sequence these genomes for phylogenetic relationships, right? So we've been able to identify genetically related species now that we can easily sequence the genomes of these organisms, right? And so a lot of these kinetoplasts are very similar in genomic background, cellular structure, but they cause very different human diseases, and they're transmitted by some different vectors, right? So early on, we talked about vectors and their role in parasitology, and they play a major role in parasitology as vehicles, right? These vectors can transmit parasitic organisms, right? These parasitic protozoa that we're talking about, they can be transmitted through these arthropod vectors, right? So we're going to look today at some human diseases that are caused by these parasitic protozoans that are transmitted through the bite of one of these arthropod vectors, right? So we're going to identify some very different human diseases that are transmitted by some different vectors, right? And so what are some examples of some arthropod vectors that we identified in our earlier lessons? Can you think of any? What's an example of an arthropod vector that we've talked about so far? Somebody give me an example of a vector. What is a vector?
It's a pathogen that transmits.
So what?
It's a pathogen.
It's a pathogen?
Yeah.
All vectors are pathogens?
No.
What's a vector? Y'all are scaring me.
It's something that carries a disease from one form to the other.
Right, right. So we're talking about a vector as a vehicle of transmission, right? So these arthropod vectors, we're talking about insects and that can potentially carry and transmit these parasitic organisms, all right? So the mosquitoes, the flies, right? Remember we talked about these arthropod vectors a little bit? So these are vehicles of transmission, all right? So we're saying today that we can identify these kinetoplasts by some of the diseases that they cause in human and the different arthropod vectors, all right? So let's jot down some quick characteristics of these kinetoplastids, all right? They're typically all motile, long, slender organisms, right? These are organisms that are gonna have various morphological forms that are gonna be distinguished by the location of the kinetoplast, okay, in relation to the nucleus. So they can be considered triple-mastigos, epimastigos, promastigos, and amastigos. And you're going to have a chance to explore that tonight and see the various morphological arrangements of these kinetoplasts in relationship to the nucleus. All right. They're going to contain a single flagellum, right? And most of them are going to develop asexually, right? So we're talking about a specific type of protozoan organism here, all right? They all are going to have a very similar morphological and genomic arrangement, similar genomes, similar morphological arrangements, long, slender. You're going to see that flagella present, but depending on where that kinetoplast lies, that's where we can distinguish our identification of these kinetoplastids, all right? Most of them are going to reproduce asexually, all right? And let's see. All right, so we're saying here that these kinetoplastids are going to cause some very different diseases in humans, but they're mostly going to be blood and tissue type diseases, all right? And again, we're talking about those that are transmitted by arthropod vectors, right? So we're talking about being transmitted through the bite of a fly or a mosquito or tick or something of that sort, right? Arthropod vectors, all right? Two species of discussion here that you've probably heard a lot about, and I've mentioned them a little bit earlier, are the Trypanosoma species and the Leishmania species. These are two very problematic organisms as they present a lot of problems in humans, okay? Let's look at some of the human diseases that are caused by some kinetoplastids, okay? Trypanosoma species, all right? You've probably heard about African sleeping sickness, okay? The scientific name for this disease is human African Trypanosomiasis, HAT, jot that down. I will ask you about human African Trypanosomiasis on your midterm, okay? This disease is caused by an infection by Trypanosoma brucei, all right? Now, morphologically speaking, this is a kinetoplastid right here, all right? We see these really dark masses of DNA here. We see this long, slender morphology. We see this flagella on the end, all right? This is a very prototypical a kinetoplast, all right? So the disease that it causes is human African Trypanosomiasis, often referred to as African sleeping sickness, all right? The etiological agent for this disease is Trypanosoma brucei, all right? I do have a couple of videos that will walk us through this disease at the end of our talk today. But let's get some important details about human African Trypanosomiasis, right? This disease is mainly affecting those in poor populations in rural areas of Africa, right? So sub-Saharan African regions, all right? This particular disease is transmitted by the bite of a tsetse fly. Jot that down. Okay, the vector is the tsetse fly for African sleeping sickness. All right, the reservoirs of this organism, of this parasitic organism, it can be either wild or domestic animal reservoirs. Humans can also be a reservoir. but I want you to be familiar with the disease, the organism, right, which is our Trypanosoma T. brucei, and then the vector, in this case, is the tsetse fly, right? This matters. You need to be able to distinguish between the vectors that are responsible for the transmission of these different organisms, all right? So this organism is going to essentially be transmitted as this tsetse fly takes a blood meal from an infected animal, right? And the organism will be taken up during that blood meal, and then this same fly, now infected, will go off and bite a human for a blood meal, and it will transmit this parasitic organism, and that's how the life cycle initiates in the human.
I got something to share, Dr. London.
Sure.
Actually, I had a friend of mine who was suffering from this infection.
Sleeping sickness?
Yes. Yeah.
Yeah. And so what were the symptoms? Did they have a very serious symptoms, or was it just like the joint pains and everything?
He just slept like
huh?
He was just sleeping.
He was just sleeping. And so and so what we'll see in another couple slides is that it essentially leads to messing up like your causing like neurological disorders, right? And they end up having a sleep disorder. Yeah, it can definitely because this is one of the few that can actually cross the blood-brain barrier. So it can get into the CNS and cause neurological disorders. And that's exactly what can happen. It causes a sleeping disorder. Absolutely. So, I mean, so I've never known or seen anyone exhibiting those symptoms. So what did that look like in your friend? Like
because here in the States, right, you can't find it over there. But if you go to Africa, like in those Goya area, you can find it. You can even see the insect that causes it. The tsetse fly. You see the insect, yeah.
So that's why you use the nets and things like that, right? To try and keep these insects away.
Yes, right? Yes.
Yeah. So yeah, no, it's definitely a real issue in Africa. Human African trypanosomiasis can be potentially fatal if left untreated. All right. And so again, this is a blood disorder. So you've got these parasites multiplying in the lymph and in our blood. All right. It's a chronic disorder. You know, you see symptoms like headache and fever and weakness, and you got these joint pains and so forth. But again, this is kind of a unique one in that it can progress or cross over into the central nervous system and cause disruptions in our neurological system. And it can lead to a sleep disorder, hence the name African sleeping sickness. All right. Well, I can imagine that this is a very uncomfortable disease to have. I mean, I think I'm sleep deprived because I have 2 small kids, but I can imagine that this right here, like my sleep deprivation probably doesn't compare to what someone over here with sleeping sickness has. Right. It's a very painful disease. It's very it can be potentially debilitating, and it can be potentially fatal. All right. Yeah. Thank you for sharing that. So these are real issues here. And I know a lot of this stuff tends to go over our heads here in the US because we don't hear a lot about it, but these are real issues globally. I mean, it's certainly a global public health concern for sure. Because I mean, in those regions in sub-Saharan Africa, there's not much that you can do to avoid them, right? So again, we're looking at the life cycle of T. brucei, Trypanosoma brucei. This is a organism that is transmitted through the bite of a tsetse fly, and it's transmitted to the it's transmitting the parasite, or the fly itself. It's uptaking the organism when it takes its blood meal, when it's feeding on these domestic animals that are infected, and it passes it on to humans when it comes over and takes a blood meal from the human, right? And so what happens is that it's injecting these organisms, right? And it migrates into our bloodstreams. It continues to move through different cycles of development, and you're gonna investigate that a little bit tonight. So we won't spend a whole lot of time on it today, but it's most certainly a blood disorder where these organisms can continue to multiply in the bloodstream and cause a lot of clinical problems. All right, so that's T. brucei, and I do have a couple of videos that we'll get to. Another human disease that's caused by Trypanosoma species is Chagas disease. You've probably heard about Chagas disease. I think you may have characterized it already at some point. But Chagas disease is the American Trypanosomiasis, right? So we looked at sleeping sickness as the African Trypanosomiasis, but Chagas disease we're seeing over in the Americas. A little bit of North America, but definitely in the South Americas. Again, characteristically, we've got this long, slender protozoan organism. We've got that flagella on the end. We've got these characteristic kinetoplasts here. All right, so we are looking at Trypanosoma cruzi as the effective agent of Chagas disease. Trypanosoma cruzi. All right, so now these are two different organisms. So again, like we're saying, in terms of genome analysis and morphological arrangements, they're very similar. But these organisms at the species level, they're a little bit different, and they cause very different diseases in humans. All right, so this disease, Chagas disease, is transmitted by the bite of the Triomatine bug. All right. It's transmitted by the bite of the Triomatine bug. And I do have a video that, again, will go through that life cycle, right? But you often hear this Triotomine bug referred to as the kissing bug, right? People get infected when these bugs basically come and feed on like you get a bug bite on your face, all right? You're sleeping, you get a bug bite, and you will become infected with this protozoan organism, all right? Humans, so the parasite can be harbored in their feces. All right, humans are infected when the parasite, if the bug feces enters their body. And what you'll see in your video, I hope I was able to find this video again, is basically the bug is going to take a bite out of your skin, on your face or somewhere, which is what the video kind of illustrates, that you get a bug bite on your face. When that bug bites you, it's going to defecate on your face, right? Somehow or another, the contents of the bug defecating will make its way to your mouth, all right? And you then become, you can also become infected, all right? So the kissing bug is responsible for transmitting T. cruzi. So two phases of Chagas disease. We can have an acute disease that's going to be last a few weeks, maybe a few months, with some relatively mild symptoms: fever, headache, fatigue, rash, right? Symptoms typically go away on their own. However, again, you can also manifest a chronic illness, right? That can occur over a 10 to 20 year period post-infection, right? And these are going to manifest itself with some very serious clinical symptoms, okay? Irregular heartbeat, congestive heart failure, difficulty swallowing, very serious chronic disease, all right? So Chagas disease is caused by T. cruzi. It's transmitted by the kissing bug, okay? Very similar life cycle where the vector here, the kissing bug, will take up the organism from an infected animal that it has bitten, and then it will in turn transmit that organism to humans also after taking a blood meal from a human, all right? Another human disease is caused, now we're looking at Leishmania species. All right, Leishmania parasite also is something of public health concern. And again, we're seeing this over in these rural sub-Saharan African areas. All right, Leishmania parasite is transmitted by the bites of an infected female phlebotomine sandfly. Okay, the disease that it causes is Leishmaniasis. Write that down. Leishmaniasis is a human disease caused by the Leishmania parasite. It's transmitted through the bite of a female phlebotomine sandfly. We're seeing it in tropics, subtropics, affects very, very poor populations. Again, here we have our very prototypical kinetoplastic organism here. We've got that slender morphology. We've got this long flagella. We've got various kinetoplasts present in this organism, right? This image right here is a picture of a phlebotomine sandfly taking a blood meal from a human. So you're basically getting bit by this sandfly, and they're going to transmit this parasitic organism into your bloodstream. Very unfortunate. Very unfortunate. Okay. Leishmaniasis can exist in 3 different forms, from being just kind of a skin disease or to actually being very potentially fatal in affecting our internal organs. All right, so write these 3 forms down. Leishmaniasis can present in 3 different forms: visceral leishmaniasis, cutaneous leishmaniasis, and mucocutaneous leishmaniasis. Okay, the visceral form of the disease is the most serious. It can be potentially fatal if left untreated. Okay, evidence or clinical symptoms of visceral leishmaniasis will manifest itself in internal organ damage. Okay, so you're going to see fever, weight loss, enlarged spleen, and liver, anemia. All right, these are very life-threatening symptoms. Okay, this is certainly evidence that your body is no longer maintaining that nice homeostatic environment. All right, cutaneous leishmaniasis is going to be present with like skin sores, right? This is the most common form that we see. So you're going to see some ulcers, a little scarring. You might get some physical disabilities, but cutaneous leishmaniasis is the most common form that we see in the population, and it's characterized by skin lesions. Okay. Mucocutaneous leishmaniasis is one of the least commonly observed symptoms or forms of the disease. It does attack your mucosa. So we're talking about the linings of your throat and your nose and your mouth. It can destroy mucosa. We don't see this very often in populations. All right. So I do want you to be able to have, again, we don't expect you to be experts when you're finished with this class, but we do want you to be able to have, be able to have some intelligent discussion about these parasitic organisms that we're discussing. If you hear, someone talking about Leishmania or Trypanosomiasis, certain diseases should come to mind. Okay, if you hear Chagas disease, you should immediately start thinking about, the vector, the organism that causes it. Okay, how is it transmitted? It's transmitted through the bite of a fly. Some very basic intelligent discussion as someone who's getting a master's degree in microbiology and you've taken a parasitology course. Parasitology, though you don't hear a lot about it in your everyday life, it is a major area of concern globally and from a public health perspective. Okay, these parasites are important because they do cause human disease. All right, so Leishmaniasis, Trypanosomiasis, I do want you to be able to identify the relationships between the disease, the organism, and the symptoms, okay, and the vector, because these are all transmitted by different vectors. They have different manifestations clinically, they cause very different human diseases. So again, 3 different forms of leishmaniasis. This is just a visual for those of us that are visual. Visceral leishmaniasis is the most potentially lethal. It's more severe. It affects our internal organs. Anything that bothers or disrupts our internal environment is not ideal, okay? Leishmaniasis, again, we're clear, is transmitted through the bite of the phlebotomine sandfly. Okay, we're seeing this affect very, very poor countries, over 1.3 million cases annually. That's a very huge public health concern. Okay, so we got visceral leishmaniasis. We've got cutaneous leishmaniasis as the most common form of the disease, and it's characterized by the presence of skin lesions. And then we have mucosal. Okay, this one is a little bit less common, but we do, see it. It's destroying our mucosal linings of the mouth, nose, and throat areas. Okay, so do take some time and review that tonight. Life cycle, again, you've got the bite of the fly. The fly takes a blood meal from an infected animal, and he's going to transmit that organism over to the human when it takes a blood meal from the human, and the life cycle will continue. All right. Another group of parasitic protozoans that we cannot overlook are the Apicomplexa. Write these terms down real quick. Apicomplexa, all right? These are our unicellular spore-forming protists, all right? They're gonna contain a special type of plastid that we'll call an apicoplast, all right? These are obviously parasitic organisms. They're typically non-motile. They can reproduce both sexually and asexually, okay? We're looking at our Apicomplexa now. These are parasitic protozoa. They're a little bit unique. They have a very complex life cycle. Okay. There are seven. Is it seven? There are 7 species that affect humans, and one of those is the Plasmodium species. All right. So the Apicomplexa, again, they have a very complex life cycle that cycles through the sporogamy, mergamy, and gametogamy. You're going to explore this a little bit tonight, again, in your extension exercise. I'm definitely going to give you till midnight tonight to get this exercise done, although it should be 7 o'clock. The organism can be transmitted by various ways. In this case, it depends on the actual organism. In this case, we want to take a look at the genera that are pathogenic, all right? The Plasmodium species are one of the 7 that cause human disease, but this one has one of the greatest impacts on human health, all right? Plasmodium causes what disease, guys? We've talked about this. What disease does Plasmodium cause?
Malaria.
Malaria, all right, malaria. So of all of the different, I want to say 7 species of Apicomplexa, these that cause human disease, these Plasmodium organisms are super serious, okay? There are more than 100 species of Plasmodium. They can infect both humans and animals, mammals, but 4 of these species in particular infect humans, and they cause malaria. Jot these down. You're going to explore them a little bit more tonight, okay? 4 species of Plasmodium. These are under our group of Apicomplexa. These are just another, again, we're just categorizing our parasitic protozoa. So the Apicomplexa, we have this Plasmodium as an example of one that is pathogenic and causes human disease. All right, it causes the disease malaria. All right, you have Plasmodium falciparum, you've got Plasmodium vivax, ovale, and malariae. All right, they all cause different degrees of malaria. All right, there's one that's more lethal than the other. Take a wild guess which one it is. All right. We're going to explore these Plasmodium species tonight for homework. All right. Real quick, some basic facts about malaria. It's transmitted by the bite of the Anopheles mosquito. All right. If caught early, it can be cured. All right. It's a very common disease in certain parts of the world. Okay. Symptoms can range from mild to being fatal. Again, depending upon which species you are infected with. All right. Characteristic symptoms are fever and chills, and you know the list can go on depending on the health of the individual, right? We talked about like the immune system and all the other factors that play a role in clinical manifestations of an infection. That is the case here with malaria, right? But some typical symptoms are going to be fever and chills. All right. This announcement doesn't apply to this class, but you do have a pretty extensive extension exercise tonight. And I'm not going to make it due by 7. I'm going to make it due by midnight. All right. You do write these questions down, and you're going to respond to 4 things that we looked at today. 1, I want you to take some time and read and be able to distinguish between the 4 morphological forms of the Kinetoplastids. Okay. The next one, I want you to describe the 3 life cycles of an apicomplexan. All right. Lastly, I want you to be able to describe how malaria is transmitted. All right. You're including the stages of the malaria parasite development. So you're going to have to do some research tonight and some reading for understanding. All right. I'm not just going to tell you today. All right, and then lastly, I want you to describe the malarial manifestations due to the infections by one of the 4 malaria parasites. So I just identified 4 malarial parasites that cause human disease. I want you to characterize the infection with one of those, okay? Those infections can look different depending on which organism you're infected with, which species you're infected with, right? So you're gonna choose one and describe the manifestation of that infection in a human. All right. So this extension will take you a little time tonight. So don't take it for granted. Do not wait till the last minute to do it. Before we go, I do have a couple videos that I would like to share to sort of tie in some of the things that we just talked about today. Go ahead and open your notes. Don't close your notes. I want you to fill in your notes where necessary. After viewing these life cycles. These videos should make it a little bit more, might make a little bit more sense and connect for you here, okay? So let's start with T. cruzi, all right? T. cruzi causing, what disease does T. cruzi cause? Hello? Which human disease is caused by Trypanosome cruzi? Hello?
Is it Chagas disease?
It's Chagas disease. Thank you. All right, so let's take a look at this life cycle and see what happens.
Life cycle of Trypanosome cruzi in the human host. In this video, we see the life cycle of Trypanosome cruzi in the human host. The trypanosome life cycle begins with the triatomine insect, which is infected with T. cruzi, biting a human host. Following the ingestion of blood, the metacyclic trypamastigotes are released with the feces from the insect near the area of the bite. Note that this form has great motility. Observe that the parasite enters the host through the bite site when the host scratches the site or when the parasite moves to a region covered by mucosa. The parasite reaches the mammalian bloodstream and subsequently invades different cell types that it encounters, such as macrophages, muscle cells, epithelial cells, and neurons. In this example, the attachment of the trypomastigote form of the parasite to the macrophage surface is observed. The process of internalization via phagocytosis begins with the formation of pseudopods. During and after the internalization of the parasite, the parasitophorous vacuole assembles. Host cell lysosomes migrate toward the internalization region and fuse with the parasitophorous vacuole. The lysosomal content is released in the vacuole. However, the parasite is not affected. In the vacuole, the trypomastigote transforms into an amastigote. This transformation is accompanied by the disruption of the parasitophorous vacuole membrane. The mastigotes are released into the cytoplasm of the host cell and divide multiple times. Observe how many mastigotes are accumulating through the division process. They can occupy the entire cytoplasm of the host cell. Following division, the mastigotes transform into trypomastigotes, which show intense and constant movement that culminates with their bursting out of the cell. The trypomastigotes reach the extracellular space and, subsequently, the bloodstream. The parasites will now infect new cells.
Okay, so very simple illustration of what's happening here. Individual got a bug bite. At the site of the bug bite, you know, the organism essentially enters the host, right? It's going to be uptaken in the host cells. The immune system is going to begin to respond. You're going to see the activity of different macrophages and so forth in an attempt to respond to it. But these guys are going to be quite resistant and they're going to continue replicating in their various stages of development. Excuse me, from the amastigote to the trypomastigote and so forth. And the clinical symptoms will begin to manifest. Let's see, I have another one. So that was TQC. Let's see, what else do I have? Oh, can I close it? Wait, what am I in Chrome? Yeah. Let's see, let's look at human African sleeping sickness.
Human African Trypanosomiasis, also known as sleeping sickness, is a disease caused by a group of parasites called Trypanosomyosis. The two main types causing human disease are Trypanosomyosis gambiense and rhodesiense. There are other types of Trypanosomyosis which cause animal disease and very occasionally infect humans. Chagas disease, also known as American Trypanosomiasis, is a different disease caused by Trypanosoma cruzi. Gambiense is the most common form of African Trypanosomiasis, accounting for about 97% of all cases. It's found in countries in Central and West Africa and tends to cause the chronic form of disease. Rhodesiense accounts for about 3% of cases. It's found in countries in Eastern and Southern Africa and causes a more acute infection. Although there were about 300,000 estimated cases in 1995, intense control activities in affected countries have led to a dramatic decline in cases. In 2014, the number of cases was estimated to be about 15,000. Let's have a look at how the disease is transmitted. The parasite is transmitted to humans through the bite of an infected tsetse fly. These flies are only found in sub-Saharan Africa, most commonly in rural areas. Both female and male flies can transmit the infection. These flies tend to bite during daylight hours. Flies can get infected when they bite a person or animal infected with the parasite. Domestic and wild animals can carry the parasite and are important reservoirs of infection, especially for Rhodesiense. Humans are the main reservoir for Gambiense. Very uncommonly, the infection can be transmitted from mother to baby, or through blood transfusions, infected needles and syringes, or sexually. Let's have a look at how the disease presents. There are two main stages of the disease. In the first or early stage, the parasite is found outside the central nervous system, mainly in the blood, lymphatic system, or other organs such as the heart, liver, spleen, or eye. In the second or late stage, the parasite invades the central nervous system. Initially, there can be a painful sore, also called a canker, at the site of the tsetse fly bite. Symptoms of the first stage include fever, muscle and joint pain, headaches, itching, and enlarged lymph nodes. It can lead to disease in the affected organs, such as inflammation of the heart muscle, enlargement of the spleen and liver, and eye disease. In the second stage, there can be a range of neurological symptoms such as changes in personality, psychiatric symptoms, weakness, or problems with movement. The disease can affect sleep. Patients tend to have nighttime sleep disturbances and are sleepy during the day. This can get progressively worse, with patients sleeping most of the time. This is why the disease is called sleeping sickness. If left untreated, the disease can lead to death. The severity of the disease and how quickly the disease progresses from the first stage to the second stage depends on the type of parasite. In Rhodesiense infections, it's quite fast, usually a few weeks. On the other hand, in Gambiense infections, it may take several years. People who get infected may not have symptoms for months, if not years. When they do, they're usually in the second stage. The disease is diagnosed by laboratory techniques, usually by examining the blood under the microscope and looking for the parasite. This usually works well for Rhodesiense infections, but harder to do for Gambiense infections. The classic method of diagnosing Gambiense infections is by examining fluid from a lymph node under a microscope. Second stage infections can be diagnosed by examining cerebrospinal fluid. Blood tests that look for antibodies against the parasite can sometimes be used. There is a test called a card agglutination test that is used for screening for people with infections. This test only works for people with Gambiense infections. PCR tests that look for the genetic fingerprint of the parasite can also be used. There's a limited range of effective medication available to treat the disease. The type of medication used will depend on the type of parasite and the stage of the disease. The disease is harder to treat once it's progressed to the second stage. The medication used is more toxic and complicated to administer. So, how do we prevent and control the disease? Let's have a look at individual measures and also measures that could be done at a community level. At an individual level, preventative measures focus on avoiding contact with the tsetse flies. These include wearing appropriate clothing that covers exposed parts of the body, avoiding bushes in areas that tsetse flies are known to live. Insect repellents are not that effective against these flies, but may prevent other insect bites that may cause other diseases. These measures may not always be possible to do for people living and working in these areas. There is no vaccine or preventative drugs that can be taken to stop people from getting the disease. Intense efforts to control the disease have been led by the World Health Organization, national sleeping sickness control programs, and others such as pharmaceutical companies and non-governmental organizations. They focus on two areas: reducing the number of people with disease and controlling tsetse flies. Reducing the number of people with disease needs good surveillance, diagnosis, and treatment that is effective, prompt, and accessible. Controlling tsetse flies is usually done by insecticide-impregnated traps and application of insecticides to the resting sites of flies. Controlling the disease in animal reservoirs is generally hard to do. And that's a quick look at sleeping sickness, a neglected tropical disease that can have a significant impact on the health of individuals and entire communities. For more information.
All right, so let's stop there. That's a lot of information, but these are very important diseases that I think, you know, we tend to overlook over here, but we need to be able to have a global perspective because we do live in a global world. And so parasitology, we cannot ignore these. All right, so I'm going to stop right here. Again, you do have a pretty extensive extension exercise tonight. I will allow you until midnight to